Okay, I guess we'd better make a start. So it's a pleasure to see you all again after a little break. And who are the people who have TP with me this week? Okay, right. So I'm sorry we have to have one session on Saturday morning. Yeah, you've seen that in your timetable. We start this afternoon and the second session has to be on Saturday. Okay, there's no way to avoid it. We have a bank holiday this week on the 11th, so there was just no way to get the third session on a weekday. So we'll just have to live with that. Okay. Right. So the next couple of lectures will be on pathogenic bacteria. So today what we are going to look at are just some general characteristics of bacterial pathogens and kind of general presentation about what any bacterium has to do if it's going to successfully infect somebody and cause disease. Hopefully then we'll have a section on host defenses against bacterial infection. And then in the next session we'll be looking at some specific examples. Okay, so with this, or rather on this subject, it's kind of useful to go right back to the beginning with Koch's postulates, which were introduced by Robert Koch in the 19th century, who, along with Pasteur, was one of the leading microbiologists who identified many bacteria responsible for specific diseases. And in this work, Koch put forward these following criteria that, according to him, allowed one to prove that a particular bacterium was responsible for a disease. So, first of all, you've got to show that the bacterium is present in all patients who have the pathology, and of course, it's absent in healthy people. Now, you should also find the bacterium in the affected organ. Okay, so the big example would be Mycobacterium tuberculosis causes a lung pathology. You find the bacterium in the lung. Okay, let's go back a bit. So you might think that's enough, okay? The bacterium is there in people who are sick, not in healthy people. It's in the affected organ. Okay, that's it. Well, it's not enough because if you show these two things, please take a seat. You've demonstrated that there is a correlation between the presence of the bacterium and the presence of the pathology. Now, correlation is not necessarily the same thing as causation. Now, this was the big argument or discussion between Louis Pasteur and members of the Academy of Medicine who didn't believe that the bacteria were causing disease. Okay, so Pasteur was able to say, oh, look, I've got bacteria here. They're in all the sick people. They're not in the healthy people. This is causing the disease. And members of the Academy of Medicine said, no, you've got it the wrong way round, Mr. Pasteur, you idiot. Right? Of course you find bacteria. These people are sick, right? They're sick. That's why they have bacteria in their diseased organs. Okay? So it's not so easy to deduce or induce causation just from correlation. So this is why Koch went a bit further. So he said, okay, what you've got to do to prove causation is you get a pure culture of the bacterium, you isolate it, and then you inoculate it into a healthy subject, probably an animal, or maybe, who knows, a volunteer, or a criminal condemned to death. I'm not sure that ever happened, but you could imagine that might be the case. And then if you do that, then you should, the bacterium should provoke the disease. You perform a controlled experiment, and that's what allows you to infer causality. Now, just the cherry on the cake here is that after you've done that, then you should be able to re-isolate the same organism from the experimentally infected subject. So if you do that, then you've really done everything to prove that the bacterium is causing the disease. So I like to present this because this is just a really nice miniature example of the scientific method. You, in the first step, okay, you've got to, collect data. This allows you to establish a correlation. And then in the next step, you perform a controlled experiment, and that's what allows you to conclude on causation. This is why, microbiology is a science, and something like history, economics, that sort of stuff, sociology, that's not a science, as far as I'm concerned. Sciences Po politics isn't a science. Ridiculous. Anyway, slight digression there. Now, ideally, if you want, this will be true for any kind of infectious pathogen, right? It's not just restricted to bacteria. The same criteria could be invoked for viruses, parasitic pathogens. And so these criteria can still be used. And ideally, for any particular infectious disease, you would want to establish each of these 4 points, all right? But it's not always true. It's not always possible. Well, one of the reasons is because, something that wasn't known about in Koch's day is the status of healthy carriers. So some people may be infected but not display symptoms. So in that case, up here, number 1 will not really be proven. Your correlation will not be perfect. So the first example of that was a lady in the 19th century called Typhoid Mary. She was persistently infected with salmonella, and she was not ill herself, but she was able to transmit the bacterium to dozens of people because she worked as a cook. Healthy carriers, you get that in quite a lot of diseases. As we'll see, maybe Streptococcus pneumoniae, that's one of the diseases, bacterial pathogens we'll be looking at. Probably Staphylococcus aureus, maybe 1/3 or 1/4 of the people here carry Staphylococcus aureus, but you're not going to be sick. So that's quite frequent. Sometimes it's not really possible to culture the infectious agent, so you can't really validate point number three. So that's true for human papillomaviruses or Mycobacterium leprae that causes leprosy. Very, very difficult to culture these agents. And of course, it may not be ethically possible to inoculate the infectious agent into a healthy individual. Okay, if your bacterium or virus can infect animals and cause disease in them, then you have an animal model. That's okay. But if your pathogen only infects human beings, then ethically you cannot prove point number three. You can't do this experiment. Okay? So for something like hepatitis C virus, only causes disease in humans. You can't actually inoculate it into people to prove that this is this is causing disease. Okay, so that's the general introduction. So once you've done that, you've got a list. You've established a list of bacterial pathogens and the diseases they cause. And what turns out to be the case is that there are two classes of pathogenic bacteria: strict pathogens and opportunistic pathogens. So an example of a strict pathogen would be Mycobacterium tuberculosis. And an opportunistic pathogen would be Pseudomonas aeruginosa, both of which could cause lung infections. Now, strict pathogens, they always cause disease and they only cause disease. You don't find them in any other context. They're only present in the context of pathology. Opportunistic pathogens seem to spend most of their bacterial lives doing something else. Generally, they don't cause disease, but in some instances they can. So, opportunistic pathogens, most strains will not be associated with disease, but some of them can cause pathology. Now, their basic lifestyle is either as commensal bacteria, so this is the most common situation. So Escherichia coli, Staphylococcus aureus, they're commensal bacteria that live in us and on us. Okay, and some strains can become pathogens. Other types of opportunistic pathogens are saprophytes. So they don't live anywhere near the human body normally. They're, chemo-organoheterotrophs, just degrading some organic material in the soil. So that would be the case of Pseudomonas aeruginosa. And one of the things that you have to try and understand in today's lecture and the next one is certainly within opportunistic pathogens in commensal bacteria, what is the difference between a harmless strain and a pathogenic strain of Escherichia coli, for example? And as we'll see later on, this is to do with virulence factors. Okay, so the harmless strains don't express virulence factors. The pathogenic strains have virulence factors. Okay. Virulence. What is that? I just used that word just now. What is it? Well, it just means it's a general term relating to the ability of an infectious agent to produce disease. So commensal Escherichia coli that we carry in our gut live there, they colonize us, but they don't cause disease. They're avirulent. And there are two main components to virulence. One is the capacity to infect or invade or colonize new hosts. So you might call this a kind of infectious potential. And the second is the capacity to harm the host, to damage the host. And this is really the pathogenic potential. So for any kind of infectious pathogen, both of these need to occur. Now you can measure these experimentally by determining the ID50, the 50% infectious dose, and the LD50, the 50% lethal dose of the microorganism. And of course, different strains of the same bacterium can have different values for these measurements. So, for example, commensal bacterium, which is harmless, you might be able to infect and colonize new hosts, but it won't have any pathogenic potential. Okay. So how do you measure the ID50 or the LD50? Very simple. You prepare different doses of the bacterium and you inject them or administer them to a bunch of animals. You might infect 10 mice at each dose or 6 or something like that. And then you follow them over time and you count the number of animals that either die or display symptoms. And this will allow you to determine the 50% lethal dose and the 50% infectious dose. Okay? So for the lethal dose, you'll be counting the number of animals that die. For the infectious dose, the number that display symptoms. And what you get is some kind of curve like this, so that as the dose injected goes up, then you'll have more and more of the animals which either display symptoms or die. Okay? If you're talking about death, then your LD50 will be where this curve cuts the 50% of dead animals line here. So here, the LD50 is going to be down here, whatever dose that is. So here are some examples of ID50s for different bacteria. So of course, okay, on this kind of curve here, right, the higher the ID50, the less infectious it is because you need a higher dose to cause symptoms. The higher the LD50, the less pathogenic the bacterium is because you need to inject more to kill the experimental animal. So you can see that, for example, this thing here, Aeromonas hydrophila, in human beings with an oral transmission route, you know, even like if you swallow 10 to 100 million of these bacteria, only 50% of people will get sick. So this is not very pathogenic, okay, not very easily transmissible. Similar here for Mycobacterium avium, by the oral route, very, very poorly infectious. Pseudomonas aeruginosa, 10 to the 10. You can swallow 1 billion of these guys. They're not going to do you any harm. So the route of infection is important because Mycobacterium pseudomonas are probably not going to live very well in the gut. They're adapted to living in the lung. So that's why these ID50 values are so high here, because we're putting them in the wrong place. Whereas something like this, Legionella pneumophila in the guinea pig, if you give this one by aerosol to the respiratory tract, okay, this is much more infectious. Fewer than 100 will be necessary to infect 50% of the experimental animals. Okay, another example where infectious route is important. So if you have trouble, if you come into contact with anthrax bacillus, if you have 30 to 50 endospores under the skin, then you'll have a 50% chance of being infected, and you'll have to inhale at least 1,000 in order to become infected. So those figures are from humans, and we know them because there was an accident in a research institute in Russia working on biological warfare. So the anthrax and bacillus endospores were released, and the people working there were exposed to them. And so it was possible to determine the ID50 for anthrax bacillus. Oh, the question's in the wrong place here. Okay, we'll come back to that in a minute. Okay, so ID50, LD50, and the idea that there's an infectious potential and a pathogenic potential. Now you can break down these processes a little bit further. So during an infection, the bacterium's got to first colonize and be transmitted to a new host. Once it arrives in the new host, it's got to be able to colonize it. Once it's there, it's got to be able to grow in the host and survive. And then in order to really cause disease, then it has to escape from host defenses, that is to avoid being eliminated by the host immune response. And finally, it needs to damage the host. So these characteristics, 1, 2, and 3, these are to do with the ID50, okay, the infectious potential. 3, 4, and 5, these will be related to the LD50, how pathogenic the infection is. So there's some kind of overlap here, right? But the first 3, these are to do with infection, and the 3, 4, and 5, these are to do with pathogenicity. So let's look at transmission first. So just like for any infectious disease, there are 2 types of transmission, direct and indirect. So direct means one infected person has got to transmit the bacterium directly to the next person along in the transmission chain. So examples are Corynebacterium diphtheriae, Mycobacterium tuberculosis. Okay. Indirect transmission also exists. Okay. So in this case, you've got one infected person. Bacteria are transmitted to something else. And then later on, they infect the next person. So there's no direct contact between these two people who get infected. So there's some kind of intermediate stage. So for Bacillus anthracis, it's the endospores in the soil. They can lie dormant for years or decades before infecting another animal. For Listeria monocytogenes, it causes gastroenteritis. It can grow on contaminated food for days or weeks in the fridge. Doesn't have to be a direct contact between you and the person who cut the slice off the pâté in the butcher's shop. Vibrio cholerae, okay, causes cholera. So here the transmission is bacteria is excreted in the feces, goes into the water, and people who drink that water can get infected. No direct contact. Okay, routes of entry. Either orally, okay, obviously bacteria that cause gastroenteritis are going to enter by the mouth. So Vibrio cholerae, the fecal oral route, frequent mechanism of infection. Respiratory, okay, so if you inhale bacteria, this can be a route of entry, or all of these things like tuberculosis, diphtheria. Okay, so there's a link between symptoms and transmission very often. Okay, so diphtheria, Mycobacterium tuberculosis, they make you cough. So this is going to enhance the spread of the pathogen. Okay, and that's what allows it to go and infect the next person, because someone who's coughing is generating an aerosol of infectious bacteria that can be inhaled by someone standing next to them. Okay, cutaneous infection. This is quite rare. Most bacterial infections are not transmitted across the by deposition on the skin, but there are some examples. So Rickettsia, typhi, and Provozekii, these cause typhus. And typhus is spread by lice, clothes lice. Okay. So what happens is the lice. Everybody know what that is? French for lice is poo, right? Okay. So the lice are infected, and when they bite you, that's not what transmits the infection. But when they're biting you, they're also going to defecate on you. And these louse feces have got a lot of bacteria in there. So when you're bitten, you're going to scratch yourself. And that's what allows the bacteria to transmit across the skin. And then you can catch typhus. So that's quite rare. Okay, not very many bacteria are transmitted this way. What is a lot more frequent is transcutaneous infection. So in this case, the skin will be damaged. Okay, you get a cut or prick yourself on a rose thorn or something like that. So cuts and scratches, they can get infected. So very, very frequent to get, streptococcal infection. You can catch Clostridium tetani from doing work outside in your garden or something like that. And last but not least, sexual transmission. Treponema pallidum causes syphilis, Neisseria gonorrhea, chlamydia. These kind of bacteria are transmitted at the genital mucosa. Okay, let's go back to the question because I put it in the wrong place. So just let's have a quick question here. True or false? Everybody thought about that? Who says true? Who says false? Yeah, you're right. Okay, it sounds reasonable, but don't forget there are dormant forms that can survive outside the host. So Bacillus anthracis, this is the perfect example. Endospores can lie dormant and they can be involved in indirect transmission. Good. Okay, so we were up to here, right? So we've got transmission, so the bacteria arrive at some kind of susceptible surface in the body, mostly at the mucosa. Now, in order to persist, the first thing they've got to do is adhere specifically to host tissues, right? And this is a function that is performed by bacterial adhesins. What are adhesins? There are two types, fimbriae, and a kind of catch-all term, which is non-fimbrial adhesins, that is everything else. So just as a bit of a reminder, fimbriae, what are they? They're filamentous structures, can be present in gram-positives and gram-negatives. So they're not flagellae, they're not the same thing as a conjugation pilus. So they're often involved in adhesion, either to inert surfaces or for the formation of biofilms. But for pathogens, they are useful for adhesion to host cells. And depending on which adhesin is expressed, this can be an important determinant of the tissue that can be colonized by these bacteria. Okay, so Escherichia coli can cause a variety of gastrointestinal infections and can also cause urinary tract infections. Now, the uropathogenic Escherichia coli express a particular fimbrial adhesin that allows them to attach to the urethra. So any kind of strain of E. coli that does not express this fimbrial adhesin cannot cause a urinary tract infection. Whereas, okay, enteropathogenic Escherichia coli will express an adhesin that allows it to bind to enterocytes in the gut. Neisseria gonorrhea, okay, there are pathogenic and non-pathogenic strains of Neisseria gonorrhea. And the pathogens are the ones that express the type 4 fimbrial adhesin that allow it to bind to the genital mucosa and the urethra. Non-pathogenic strains, they don't express this and they'll just be washed away. Okay, non-fimbrial adhesins, what are they? Well, some kind of strains of E. coli have expressed non-fimbrial adhesins, or this is enterotoxigenic strains. Another example is Yersinia enterocolitica, once again causing a gastrointestinal infection. So the adhesin here is called YAD-A. And if you look at a strain that's knocked out for the YAD-A gene, this is what you see. So this is the bacterial cell wall. It's quite smooth on the surface. YAD-A positive strains have got these kind of structures here on the outside. Okay, so that's the adhesin, YAD-A, what it looks like. Okay, so the bacterium has been transmitted. It's bound to the host cell. The next thing it's got to do is grow in the host. Now, you might think that being in a human body is like the bacterium has died and gone to bacterial nirvana. All right, there's loads of sugar there, there's loads of amino acids, there's loads of everything. It's a very rich environment. Okay, and that's true for most things. But there is almost no free iron in extracellular fluids. So this, and as you know, this is one of the oligoelements that's necessary for bacterial growth. So this is going to be a limiting factor for the growth of pathogenic bacteria. So they all have to express some kind of system for recovering iron in the host. Now there are different ways and means of doing this. So the first is to express a very high affinity siderophore. So in this case, the siderophore will be able to pick up the very low concentrations of free iron that are around. So Mycobacterium tuberculosis does this, and Yersinia. Now the other solution is not to express a siderophore, but to exploit the fact that, in fact, iron is being transferred around inside the host with these iron transport proteins, lactoferrin, transferrin, ferritin. So if the bacteria can bind one of these proteins and import that, then they can get the iron from the host proteins. So that's what Neisseria does. So TBP, transferrin binding protein A, transferrin binding protein B. So they'll pick up the host iron transport proteins and use that as a source of iron. And the other, of course, big source of iron in your body is hemoglobin, right? So if bacteria can lyse red blood cells, they'll release hemoglobin and be able to pick up iron from that. So hemolysis, right, that can be provoked by pathogenic bacteria. They're not doing this just to really kill us. You know, they don't really want to hurt us. They just need iron, poor little things. Okay, so that's why they're doing this. Okay, so who uses hemoglobin, Vibrio cholerae, other species of Yersinia, and Pseudomonas aeruginosa? Okay, so most bacteria that arrive at a susceptible site will not be able to colonize us because they will be eliminated by host defenses. And one of the main, well, we'll talk about that in a couple of minutes, but one of the most important aspects of host defense is phagocytosis and destruction of bacteria by neutrophils and macrophages. So one of the big strategies that the pathogenic bacteria use is to try and avoid phagocytosis, to try and escape from being eaten. And this is very often the function of the capsule antigen. So Neisseria, Streptococcus, Haemophilus influenzae, these are all bacteria that express capsules, that have a capsule, and strains that do not have a capsule are generally less pathogenic. Now, the other possibility is that the bacteria will not avoid phagocytosis, but they'll have a strategy to survive even after they've been internalized. So either bacteria will escape from the phagolysosome and go and live in the cytosol. Listeria does this. Rickettsia does that. Enteroinvasive E. coli also does that. And the other strategy is to survive inside the phagolysosome. So Salmonella and Mycobacterium tuberculosis. So they prevent the phagolysosome from maturing and turn it into a kind of nice little home sweet home for bacteria. Now, another step that is also important in pathogenicity is the expression of enzymes that will damage the host. Okay. So one aspect of this is expression of collagenases, hyaluronidase, or elastase. And what these have in common is they will all attack the extracellular matrix. So here's the epithelium. Okay, could be genital mucosa, could be respiratory mucosa. And this is the site of colonization, right? So the bacteria arrive here and will start to colonize the apical pole of these epithelial cells. Now, in order to become an invasive infection, what will tend to happen is these bacteria, okay, these cells here that are infected are going to start to die, and bacteria will gain access to the basal layer. Now, in order to be able to start making a bacterial colony here, what they have to do is degrade the extracellular matrix here, okay? So that's the role of these enzymes, collagenase, hyaluronidase, elastase. So the extracellular matrix will be degraded, and that will allow these bacteria to grow in a colony. That's a very important part of invasion, okay, expressing these hydrolytic enzymes. Other hydrolytic enzymes that can cause problems are the expression of coagulase or DNase. Now, DNase, why is that important? Okay, what can happen is that once these epithelial cells are killed, then their chromosomal DNA will be released. So as you know, high molecular weight DNA, it's a very, very big molecule, it's very viscous. So it's going to form a kind of big, horrible, messy gel, and these bacteria can be stuck in there. So if they express the DNase, then all of this host cell DNA that's been released will be degraded, and then that will help them to spread from the initial site of infection. Otherwise, they're going to be caught up in this kind of DNA, blob. Coagulase, this is kind of different thing that's happening here, okay? So other bacteria, Streptococcus, produce streptokinase, all right? So streptokinase will degrade a clot. Okay, let me go and make the link here, okay? So DNA from host cells can tie up the bacteria. Now, another thing that can prevent bacteria from spreading is coagulation, right? Forming a blood clot. This will have a tendency to localize bacteria. So some bacteria produce enzymes that degrade a blood clot. Streptokinase, Streptococcus will produce this enzyme. That once again allows them to disseminate. Now, Staphylococcus aureus does something very different, does the opposite, right? It's going to induce the formation of a clot by expressing coagulase. Now, why on earth does it do that? Okay. Okay, this is going to be a blood vessel now. And you've got some Staphylococcus aureus floating around here. If they bind to the endothelium somewhere, they express coagulase, and this will induce the formation of a clot. Maybe not even in the blood vessel, maybe on the other side. Okay, they're on the other side, maybe causing some damage. They'll form a clot. So why do they do that? This is the first step in the formation of an abscess. So an abscess is this kind of like thing filled with bacteria and host cells that are trying to get in there destroy them. So you have the formation of an abscess where you've got bacteria, a blood clot, and then around the outside you've got host cells that are trying to get in and kill these bacteria. But abscesses can be very, very persistent, very difficult to get rid of. If you've ever had one, then you know that if you don't treat them, they can stay there for weeks or months. So this is why Staphylococcus aureus expresses a coagulase. It's the first step in forming an abscess, and that's the kind of strategy for persisting, even though cells of the host immune system are trying to migrate towards them and phagocytose them. So that's a very, very specific aspect of Staphylococcus aureus pathology. It was first isolated from an abscess from a patient in Aberdeen, in fact. So Staphylococcus aureus is Scottish. Okay, so expressing enzymes that would directly attack host tissues. That's one aspect of pathology, of harming the host. And the final thing that many bacteria do is directly harm the host by expressing bacterial toxins. Now, there are two types of toxins: exotoxins, which are secreted proteins, or exo, external from the bacterial cell. And the big classical example is, for example, cholera toxin, which is an example of an AB toxin. So there are two subunits. B is for binding and A is for active or activity. So Clostridium tetani, this is going to be tetanus toxin. Bacillus anthracis, they have several ones. They have names like lethal toxin and stuff like that. Okay, so how do they work? What is happening here is that this is the dimer, the AB toxin, will bind to the host cell through the binding subunit B. So this is internalized into the cell. It gets transported into the cytosol, and then the A subunit can do something that will directly manipulate what is happening inside the cell. So for the cholera toxin, the A subunit is an adenylate cyclase. It produces cyclic AMP, which will then activate the chloride, the CFTR chloride channel. So chloride is going to be actively exported into the lumen of the intestine. And in order to maintain osmotic balance, then sodium ions and water will follow the chloride. And this is what causes the characteristic watery, very copious diarrhea that is characteristic of cholera infection. Okay, so the toxin here is very directly responsible for the symptoms. And so that's why many vaccines are based on inactivated bacterial toxins. So you can develop antibodies against the toxin, and then you can get infected, but the toxin will not be active, so you won't get sick. That's not actually true for the cholera vaccine, but it's true for the anti-tetanus vaccine. Diphtheria, I'm not so sure, but okay, tetanus vaccine is based on the toxin. As well as that, endotoxins are something different. So endotoxins, this is LPS, lipopolysaccharide, in the outer leaflet of gram-negative bacteria. So exotoxins can be secreted by gram-positives or gram-negatives. Endotoxins, gram-negative only. Now, what happens here is that the endotoxins don't really provoke a very, very specific effect. They provoke systemic effects, because they are detected by elements of the immune system. So endotoxins from different gram-negative pathogens will all have a similar effect on the system. So it's going to be fever, you can have low blood pressure, hypovolemic shock, And in the end, if if this is not controlled, then endotoxin can cause septic shock and people can die from it. So one of the very, very frequent exam questions that we love to ask is endotoxin and exotoxin, what are the different, compare and contrast, this kind of thing. So exotoxins, proteins, endotoxin, LPS. Exotoxins, gram-positive and gram-negative. Endotoxins, gram-negative only. Exotoxins, different effects that are directly related to the pathology. Endotoxins, relatively non-specific effects. This is the kind of thing that we will want to hear from you in an exam answer. Okay, so we've gone through all the different steps that allow bacteria to induce pathology. So in order for bacterium to be pathogenic, they'll have to express adhesins. They'll have to have some kind of mechanism to get iron from the host. They will need to express hydrolytic enzymes in order to invade host tissues and produce toxins to actually directly harm the host. So it's not just one gene that's going to be involved here that will differentiate a harmless commensal bacterium from a pathogenic bacterium, okay, because you need all these things. And each of these systems is complicated, all right? So if you remember the biosynthesis of fimbriae adhesins for chaperone usher fimbria, you'll need chaperone protein, usher protein, the major filament protein, and the adhesin. So you need at least 4 proteins here, okay? These would all be coded on the same operon in the bacterial genome. 4 genes all coded together, all regulated together. The same will be true for siderophores, okay? So you're going to need the enzymes that produce the siderophore, and you'll need to have the ABC transporter that allows the siderophore iron complex to be imported. Once again, you need several genes. For toxins, you need at least A and B subunits of the toxin, so at least 2 genes. So say for each of these functions, we're not talking about one gene, we're talking about an operon, okay? So within the operon, you'll have all the functions that you need, or all the genes that you need for one of these functions. So that's what we're looking for. So where are they coded on the bacterial chromosome, these operons that confer pathogenicity? Well, it seems like they are found, well, sometimes they're found on plasmids, but most of the time they are in the chromosome, and they are found in pathogenicity islands. What are the characteristics of a pathogenicity island? Well, simplest thing is to look at the GC content. So this is going to be the bacterial chromosome, and then you have a region which has got lower GC content. And once you look at the edges of this region of low GC content, sometimes you can find direct repeats, which have been formed during the insertion of this DNA. So this low GC content, this is a characteristic of a mobile element of DNA. It gets transferred from one bacterium to another. The direct repeats are the borders of this insertion, and often you find an integrase present in the pathogenicity island. So this is what is inserting all of this DNA sequence. And inside you'll have V1, V2, V3, V4. These are the different genes in an operon that encode for some kind of virulence function. So it will be the different elements of a siderophore for iron, or it will be the different proteins that make up an adhesin. Okay, so mobile elements of DNA, either with some kind of mobile integrase system, or they can be bacteriophage. So in Salmonella, for example, there are different pathogenicity islands, and each one carries the genes that code for one function, okay, SPI1. got all the genes necessary for invasion of non-phagocytic cells. SPI2, it's got all the genes necessary for surviving inside the phagocytes. SPI3, it's got a siderophore system. And SPI7, this is coding for the enzymes that produce the capsule. So each pathogenicity island will carry the genes for one function involved in pathogenicity. Okay, so that's the summary so far. Strict pathogens versus opportunistic pathogens, LD50, and then these different elements, different steps in the pathogenic process that all bacteria have to perform in order to really hurt us. Okay, so let's have a kind of little break here. I'm going to send you out a clicker question. So that should be the question if you connect here. Hey, that's wrong. I got the wrong question. Oh, damn it. Not the same questions. Okay. Oops. Okay, let's see what the real questions are, because I think I sent out the one that's not really the same. Yeah, it's the wrong question. Why didn't anybody tell me? Because I was looking at, I was thinking, okay, well, this is the real question that got sent out. So uh, this one, first one is you need to. Oh, yeah, it's the wrong one. Sorry. Oh, yeah, yeah. Actually, I'm not going to. Okay, well, did everybody get this right? Okay, we already had this question before. Somebody said yes, and we just asked it before in class. So this is a bit strange. Okay, this is right. Yeah, a strict pathogen always associated with a pathological pathogenic concept. That's true. That's false. That's you're right. Okay, that's also not true. This is just a correlation. Okay, then you have to perform the controlled experiment to prove. So this is the Cox postulates. Now this one, bacteria with a high pathogenic potential have got a high LD50. That's false. Okay, good. Everybody got that right. If LD50 is low, that means you have a high pathogenic potential. Okay, sorry for sending out the wrong question. So let's think about these questions now, okay? So let's do this by a show of hands. All right, capsule antigens. They're important because they mediate adhesion. Who says yes? Who says no? You're right. It's not the wrong. B. Who says yes? Who said no? You're right, okay? Siderophore or something else. C. We just answered that one. Okay. D. E. Who says yes for E? Yeah, you're right. Gram-positive and gram-negative secrete exotoxin. D. Okay, if a pathogenic bacteria loses the virulence factor coding for an exotoxin, its ID50 will increase. Who says yes? Who says no? People who said yes, why? Because you think it's the same as this, right? Okay, so if they lose a gene, they become less pathogenic. The person who said no, why? Yes, yes. So for exotoxin, this is going to be involved in directly causing pathology, harming the host. It's not involved in transmission or being able to infect a new host, okay? So probably the ID50 will stay the same. Yeah. Ah, okay. Maybe. So it won't be able to provoke symptoms. And that's what you detect to determine the ID50. Yeah, maybe. You might be true. But the idea of this question is to get you to think about, okay, difference between LD50, ID50. So the LD50 will definitely increase. You need more bacteria to kill the infected animal. The ID50 might not change very much, but it depends what you use to detect the infection. If you're just talking about symptoms, it might also increase. If you talk about detecting bacteria with plating them out, detecting them microbiologically, then it won't change. Do you see what I mean? Okay. Okay, let's think about this ID50 experiment, right? That's a mouse. You inject the bacteria, right? Or administer the bacteria. So later on, what are you looking for the ID50? Symptoms or count bacteria? in the lung, in the intestine, on the skin, wherever the infection is. Okay, so if you use this for the ID50, if they have an exotoxin or not, probably won't change, right? Whereas if you use symptoms to determine the ID50, then maybe you're right. If they have an exotoxin, the ID50 will be lower because you'll need fewer bacteria to cause the symptoms. Okay, yeah, okay. So you lose the exotoxin, the ID50 will be higher because you need more to provoke symptoms. Yeah. If they have the exotoxin, you need less. You need fewer. Yeah. Okay. Right. Thank you. Right. Thank you for that interesting discussion. I think we have time to carry on, look at host defense a little bit. Okay. Host defense. Now, what does that involve? Most of the time, you think about the immune system. You're thinking about antibodies, cells of the host immune system that are going to eliminate bacteria. But before that, there is a lot of host defense, which is, I don't know how you could call it, not exactly passive, but really doesn't involve specific antibacterial immune effectors. So there are two types of things that I want to talk about here. Is that first is the physicochemical barriers, and then the endogenous bacterial flora. So physicochemical barriers, that could just be like the skin. Okay, it's not a very easy environment for bacteria, or pathogenic bacteria at least, to colonize. So very few bacterial infections are transmitted by cutaneous route. Mucus, okay, as we said, pathogenic bacteria have to adhere to the surface of the epithelial cells. But if this is covered by mucus, then the bacteria can't get access to the cells, okay, so they won't be able to adhere. And together with this, you have a kind of flow of the mucus over the over the mucosal surface. Right, so if you think about your respiratory mucosa, all of the cilia are beating in the same direction, so there's a flow of the mucus out of the lungs up into the upper respiratory tract, into the mouth. So this is going to expel bacteria that are caught in the mucus. And you have the same kind of thing with peristaltism. So bacteria aren't hanging around in your intestine. They're continually getting moved through the system, so they can be expelled. Same thing with urine, all right? You have a flow of urine through the urethra, and this will have a tendency to expel the bacteria that might have come into contact. So this is a very, very simple system, and that accounts for the fact that, for example, urinary infections are 14 times more frequent in women than in men, because the length of the urethra is longer in men, right? What else have we got for flow? There's saliva. Okay, that's good. You produce saliva and then it gets swallowed and bacteria are going to be destroyed in the stomach. Yeah, also urinary infections, the pH and the content of the chemical makeup of urine is not very conducive for bacterial growth. It's going to be acidic, you have uric acid, urea. These can be toxic for many bacteria. Low pH, the vaginal mucosa, very acidic medium. That's because of cooperation between the host. So glycogen is secreted, and this is used by Lactobacillus acidophilus, part of the normal vaginal bacterial flora. So this gives you low pH, which will prevent growth of many bacteria. What else do we have here? Okay, talking about flow of air. This diagram is not really very good, all right, because it shows, well, okay, this is for the intestine. But when you breathe in through your nose, the flow of air is not smooth. You don't have a you don't have a tube just going like this from your nose that goes to the trachea. Okay, in the upper respiratory tract, you've got a whole load of complicated tubes here that go up and down like this. So when the air is flowing in through your nose, you don't have smooth laminar flow, you have turbulent flow. Okay, you've got the air is going to be turbulent here. And the net effect of this is that particles that might be inhaled are going to be deposited on the surface of the upper respiratory airways. Very, very few are going to get down into the larynx or in your lungs. So this kind of defense, this kind of arrangement is what's stopping you from getting pneumonia most of the time. Okay, so most of the infections that you're going to get this winter, 99% of them in everybody are going to be upper respiratory tract infections. Lower respiratory tract infections are very rare, and that's just because of this passive or defense mechanism, just the way the upper respiratory tract is organized. You don't generally think about that when you're thinking about defense of the host, but that's really part of it. So another thing that is protecting us is our endogenous bacterial flora. Okay, so our skin is colonized by bacteria, Staphylococcus epidermidis. Our mucosal surfaces in the mouth, we've got loads of bacteria. In the gastrointestinal tract, in the gut, lots of bacteria. And these have an important role in preventing colonization by pathogens. Two mechanisms, basically. One is just quite passive. Okay, they're occupying all the niches on our skin, in our guts. So there's no space available for other bacteria to come in and colonize us. That's quite passive. The other thing is they're active. Our commensal bacteria secrete proteins that are toxic for other bacteria. So Escherichia coli secretes proteins that are called colicins. And other bacteria, Streptococcus, Bacillus, Staphylococcus, they all secrete bacteriocins, proteins that are toxic for other bacteria. So they actively prevent colonization by pathogens. Okay? So that's one of the problems. If you get treated by antibiotics, then it can disturb your gut bacteria. And that can make it much easier for you to get colonized by a pathogen, because you've taken away all of the endogenous flora, or weakened it, and that is protecting you from colonization. So one of the big problems that occurs in a hospital context is these people who are on antibiotic treatment can easily become infected by pathogenic gut bacteria. And if they're resistant, then it can be very, very difficult to treat them. So one of them is Clostridium difficile. And it has been proposed, the idea was, okay, so if our endogenous bacteria are so great, they're protecting us all the time, and people who are treated by antibiotics, they don't have any endogenous bacteria, maybe we can put some back in, and that will have a therapeutic effect. And so that's the reasoning behind what's called a fecal transplant. Okay, so the idea is you've got a healthy person here with a healthy gut bacteria, and there's somebody here who's got a Clostridium difficile infection with antibiotic-resistant pathogenic bacteria that are causing a lot of problems. So what we do, we take the feces from this person here, put it in a blender, and prepare some kind of bacterial suspension, and then administer it into this poor person's gut, either through the nose and start seeding the bacteria into the small intestine, or through the anus up through a tube here, and you can seed the bacteria into the colon. Now this is what some people try and do at home. Don't try this one, just with a kind of like enema sort of thing. You'll only be able to reach your rectum, so it's not going to work very well. So this is the scheme here. Now it might look rather distasteful, but does it work? Okay, that's the question. Does it work? Or are these like gastroenterologists just a bunch of perverts? Well, I don't know about that. So here's the results from some experiment, a clinical trial. So these are patients who've got Clostridium difficile infection, very difficult to treat with antibiotics. Treating them with vancomycin, you get 30% cure rate. And then these are the patients who got a fecal transplant. After just one treatment, 80% are cured. After, I don't know how many, but over the whole course of the therapy, more than 90% cure rate. So it's very effective. And this is something that's been developed. So this is from 2013, this paper. So this is a therapy that's been developed for these difficult to treat gut infections. Okay. Okay, I'm just going to do one more slide and then we'll stop, okay? Okay. Okay, so those are the defense mechanisms, physicochemical, endogenous bacterial flora, and now we're on to the third one, the immune system. So in the immune system, one of the ways to do this is think about innate and adaptive immune responses. So that's what we're going to do. We'll start with innate immune defenses against bacteria. And we're going to talk about soluble effectors floating around in your extracellular fluids, and then cellular defenses. So the first one, probably everybody knows, is lysozyme. You know, in your mouth, in your tears, in your extracellular fluid, in eggs. So it's an enzyme that will degrade the repeating structure of peptidoglycan. So it's going to break this beta-1,4 bond between N-acetylglucosamine and N-acetylmuramic acid. So this glycosidic linkage is only present in bacteria. It's not in any of the host structures, so it's specific. Now, of course, lysozyme on its own will only be able to degrade gram-positive cell walls because it's an enzyme and it can't get across the outer membrane of gram-negative bacteria. Also, if the gram-positive has got a capsule, it will be very difficult for lysozyme to diffuse across the capsule polymer and gain access to the peptidoglycan. So another soluble mediator of antibacterial defenses is the complement system. Okay, it's not really the place to go through the whole complement pathway. That's a job for immunologists, I guess. What I want you to know about this, at least anyway, is that the different pathways for activating complement will all converge on the C3 convertase enzyme. So C, okay, it's complement. You've got a whole cascade of proteins, C1, C3, C5. It's like the blood clotting cascade, a little bit, okay? So it's a proteolytic cleavage, and then you have another enzyme. It's activating the next one, this kind of thing. So they converge on the C3 convertase. So C3 convertase is going to cut C3 into two components, C3a and C3b. Okay, C3a and C5a, these are chemotactic proteins. They're soluble, they'll be released, they attract neutrophils. So they're going to attract cells to the site of the infection. So that's one important function of complement. Second one is C3b. So C3b will covalently bind to the surface of the bacterial cell and will recruit different complement components that form the membrane attack complex, the MAC. So the membrane attack complex is going to punch holes, form pores in the outer membrane of the bacterial cell. So that's very useful because that means that complement and lysozyme will work together. Okay, so complement on gram-negative bacterium, the membrane attack complex will form pores, and this will allow lysozyme to get access to the peptidoglycan, and together they'll be able to kill the bacterial cell. Okay. Now, so that's what complement is doing. Okay, attracts neutrophils and will form pores in the bacterial outer membrane. How is it activated? Well, the classical pathway, this is antibodies. The Mannan binding lectin, so this is a kind of innate immune receptor, will recognize some kind of let me see what this is. Okay, glycosylated structures on the outside of the bacterial cell, and it will activate complement. The alternative pathway, this is also quite interesting, but I don't know if I'm really going to tell you about that. No, not enough time. So you don't really need to know about this. Antibodies and somehow recognizing the differences between the outer surface of bacteria and the host cell, these two. And the last thing we need to know about today is antimicrobial peptides, cathelicidins and defensins. So these are short peptides. So the cathelicidins, this is alpha helix, defensins, beta sheet. Relatively short, okay. And neutrophils express them both. But the cathelicidins are otherwise in the respiratory epithelium and expressed by alveolar macrophages. So cathelicidins in the lung, defensins in the gut, okay, intestinal epithelium. So both of these antimicrobial peptides have an activity against gram-negative outer membranes. So once again, they're going to permeabilize the membrane of bacteria and allow lysozyme to gain access to the peptidoglycan. Okay. Right, that's about it. So I think we'll stop there for today. Time is just about up. So this evening, 7 p.m., I'll send out another microbe of the week. Is that okay for everybody? Or would 8 p.m. be better? Because we're in TP till 6, right? Maybe 8 p.m. tonight, okay? Gives everybody time to get home. All right, 8 p.m. for microbe of the week.